Hello friends, today I will discuss the very important cells of the nervous system, that is the neuroglia. As I told in my previous video that there are two kind of these cells, one is the neuron and second is the neuroglia. So today I will focus on the neuroglia, so which you can study in detail in my book Human Anatomy and Physiology, published by E. Redon. Okay, so let's start. So when we say neuroglia, they are also called as the glia or the glial cells. So you can say these are the specialized cells of the nervous system. Now what is their major function? So they provide the support, they provide the nourishment, they provide the protection to the neurons. So neurons are the main cells and they are the support cells. They are much smaller in size as compared to the neurons. But important thing is their number is not less, number is very high. You can see 5 to 50 times more than the neurons. And moreover, these cells can divide and multiply, but these cells cannot generate or propagate action potential. This property is only with the neurons, not with the neuroglia. So now, I will discuss what are the different kind of neuroglia. So if I discuss the central nervous system, there are 4 neuroglia. Like first is the astrocytes, then oligodendrocytes, third microglial cells, and fourth is the ependymal cells. And if we discuss in the peripheral nervous system, then we are having the neurolimocytes that are also called as Schwann cells. Then we are having the satellite cells. We will discuss all these cells one by one. First of all, coming down to the astrocytes. So astrocytes, astro mean star, site means cell. So these cells are having a shape of the star-like structure. So that means if this is the shape, you can see star-like shape. Okay, astrocytes. So many processes are present, many processes. Okay, so star-like shape. So what is the functions of astrocytes? Number of functions are there, like they help in the metabolism of neurotransmitters. They maintain the concentration of potassium ions. They help in the development of the brain. They also help in the formation of blood-brain barrier. Moreover, because of this property, that means it helps in the formation of blood-brain barrier. So the things that can enter inside the brain, they are regulated. Not all the substances are allowed to enter in the brain. This is the property of the blood-brain barrier. So you can say it regulates the entry of substances into the brain. So you can say these are the functions of the astrocytes inside the brain. Coming down to the another one, oligodendrocytes. When you say oligo means few. Dendro related to the branches, like to the tree. Okay, cytes means cell. So these are the cells in which there are few processes which are attached. Not many, but few. Okay. So if you compare the oligo and astrocytes, this is the one difference. Few processes in oligo and many in the astrocytes. Second, they are smaller in size as compared to astrocytes. Now few processes are attached to the cell body. Functions, they provide support to the neurons and they help in the formation of myelin sheath in the CNS. I will discuss this issue later on also, but remember the formation of myelin sheath around the axons within the CNS in the brain is done by the oligodendrocytes. Third, very important cells, ependymal cell. We will discuss these cells further also, but I am just discussing what are these cells. So these are, you can say, as you can see the shape, these are ciliated, cilia is present, columnar epithelial cells, columnar epithelial cells are present. So where these are present, they line the brain ventricles. We will discuss all these topics one by one. Okay. So the ventricles are present, and their function of ventricle is to form the cerebrospinal fluid. Okay, so the internal lining of the brain ventricles is because of the ependymal cells. So as I told, because ependymal cells, they line the ventricles and they help in the formation of an important fluid in the CNS, which is called as cerebrospinal fluid. Fourth one, microglial cells. Micro stands for small, glia adhere. So these are very small cells. You can see they are small phagocytic cells present in the central nervous system. So you can see these are actually the macrophages of the brain. You know, macrophages are present inside this body, and depending upon the region of the body, they are named differently. If these macrophages are present inside the brain, they are called as the microglial cells. And they function, they provide protection, okay, by engulfing any microorganism because of the phagocytosis. Coming down to the, you can say the cells of the glial cells present in the peripheral nervous system, starting with Schwann cells. So I will discuss the structure of these cells in detail. Let's say this is the axon. This axon is surrounded by a layer which is called as the myelin sheath. This myelin sheath is non-living in nature. Okay. This myelin sheath is further encapsulated by a cell. This is the living layer, living cells, and which has the cytoplasm, which has the nucleus, and this is because this nucleus is called as nucleus of Schwann cells. And both cytoplasm and Schwann cells, they are called both comprise to form the Schwann cells. And this lining, outer lining of these Schwann cells, they are called as neurolemma. So coming in the written form, Schwann cells, they are also called as neurolimocytes. Axon is surrounded by two layers. Inner layer is made up of myelin sheath, which is non-living. The outer layer, which is also called as neurolimocyte, it is living layer, and it surrounds the inner layer and contains the nucleus and cytoplasm. And the outermost layer is called as neurolemma. So now what are the functions of these cells? They help in the formation of myelin sheath around the axons in the peripheral nervous system. I just want to compare that if the myelin sheath in the central nervous system is formed by oligodendrocytes, but the myelin sheath in the peripheral nervous system is formed by these Schwann cells. Plus, another thing, if this myelin sheath or the axon is ruptured, okay, then that is regenerated by these cells. So helps in regeneration of the injured axons and the myelin sheath. So I'm just comparing oligodendrocytes and Schwann cells. In the CNS, oligodendrocytes produce myelin sheath, and only myelin sheath is present. Neurolemma is absent. But if in the peripheral nervous system, Schwann cells produce myelin sheath, and myelin sheath is present, and neurolemma is also present. Now in the upcoming video, I will discuss the coverings of the brain, including the meninges. Thank you.